guys, this is Malinki. Welcome back to my channel Voice of Malinki. Today we will talk about C4 cycle. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So first of all, one thing is very important to note that C4 pathway is not the alternative of C3. C3 pathway occurs in all plants since C3 pathway is the only process that makes sugar during photosynthesis. So every plant has to go through C3 cycle in order to produce glucose. Then what is C4 pathway? In last lecture we have talked about photorespiration. In that lecture we have seen that photorespiration is a harmful process because in this pathway there is neither synthesis of sugars nor of ATP and NADPH. Rather, it results in the release of CO2 with the utilization of ATP. Some plants which are found in dry regions like sorghum, maize, etc. go through the C3 pathway in a different manner. These plants strictly avoid photorespiration. They have a special type of leaf anatomy which is absent in those plants who do not undergo C4 pathway. The leaves of these plants have some large cells called bundle sheath cells. These cells surround vascular bundles where mainly xylem and phloem are present. The bundle sheath cells form several layers around the vascular bundles like these. These cells have very thick walls like these. Hence, they do not allow any gaseous exchange. Around bundle sheath cells, mesophyll cells are present. In these mesophyll cells, CO2 enters via stomata. In these plants, mesophyll cells lack Rubisco enzyme. Therefore, CO2 does not get any scope to bind Rubisco and to continue C3 cycle. Here, a CO2 acceptor is present in the cytoplasm called phosphoenol pyruvate or PEP, which is a 3-carbon compound. And the enzyme PEP carboxylase or PEPcase is responsible for the fixation of CO2 with PEP, which yields oxaloacetic acid, which is a 4-carbon compound. Since this is the fourth stable product in this pathway and this is a 4-carbon compound, it is called C4 pathway. Oxaloacetic acid then moves to the chloroplast stroma from cytoplasm and forms 4-carbon malic acid in the mesophyll cell itself, which is transported to the chloroplast stroma of the bundle sheath cells. Here, this malic acid is broken down to release CO2 and pyruvate. Since the wall of bundle sheath cells are very thick, they don't allow any gaseous exchange. CO2 does not move to the surrounding mesophyll cells. Rather, enters C3 cycle in the bundle sheath cell itself since Rubisco is present in bundle sheath cells. And this pyruvate is transported back to the chloroplast stroma of the mesophyll where it is converted to phosphoenol pyruvate or PEP again, thus completing the cycle. Phosphoenol pyruvate exits chloroplast and enters cytoplasm to initiate new cycle. So it is important to note that in these plants, C3 occurs in the chloroplast of bundle sheath cells, instead mesophyll cells. The bundle sheath cells lack PEP case. Hence, this CO2 does not get any scope to bind PEP case. It has only one fate to join C3 cycle. Thus, the basic pathway that results in the formation of sugars, the Kelvin pathway or C3 pathway, is common to C3 and C4 plants. In bundle sheath cells, light reaction does not occur. Therefore, oxygen is not produced in these cells. Light reaction occurs in mesophyll cells. From mesophyll cell, oxygen gets released via stomata. But this oxygen is not utilized by rubisco to undergo photorespiration, since rubisco is absent in mesophyll cell. Therefore, C4 pathway is a wonderful technique which eliminates photorespiration, the harmful process of plants.